Isso. Ju, segura esse aqui para nós. Bora, gente, começar. Bom, boa tarde a todos. Estamos aqui para mais uma edição dos Diálogos Ilum, e é um grande gente começar. Bom, boa tarde a todos. Estamos aqui para mais uma edição dos Diálogos Ilum, e é um grande gente. Sorry for the repetition. Boa tarde. Estamos aqui, então, para mais um Diálogos Ilum, e é um prazer apresentar para vocês o professor Dean Hesterberg, que eu conheço faz pouco tempo, mas sou super fã. E ele está no LNLS. When did you come to LNLS? Like 3 years ago? Yeah, 21. So Dean came to work at Sirius first as an advisor to the director in the... Voltando, eu vou fazer a introdução em português, porque depois ele vai falar em inglês, mas aí a gente vai mediar perguntas, tá? Então, só apresentar. O Dean está no CNPEM desde 2021, onde primeiro ele era um conselheiro do diretor do LNLS na parte de ciências do solo, e ele agora, além de ser advisor, ele também é pesquisador vinculado ao LNLS, né, na área de ciência dos solos. O Dean veio para cá depois de se aposentar como professor em Química de Solos, no Departamento de Solo e Ciências do Solo da Universidade Estadual da Carolina do Norte, lá nos Estados Unidos. Ele fez a graduação em Plantas e Ciências do Solo na Universidade do Sul de Illinois, e mestrado em Agronomia na Purdue University, e um doutorado em Solo e Ciências do Ambiente na Universidade da Califórnia em Riverside. Ele também trabalhou na Desculpem, retornando. Então, antes de ir para a Universidade da Carolina do Norte, ele trabalhou na Chevron, no Instituto de Pesquisa da Chevron, que é uma petrolífera, né? Grande rede dos Estados Unidos, e também no Instituto para a Fertilidade do Solo, na Holanda. E espero que vocês gostem da apresentação. Ele vai falar devagar, vai apresentar várias animações e ele quer que vocês interajam. Então, se alguém quiser fazer qualquer pergunta ou comentário, levanta a mão, aí eu e a Juliana vamos mediar aí as perguntas, tá bom? Vamos ficar provocando vocês também. Então, é um prazer. Welcome, Dean.
Muito obrigado, Ana. And muito obrigado a todos para participar neste seminar. Sinto muito, mas eu vou falar em inglês, ok? É melhor será maior para vocês e para mim. Mas quero que vocês tenham muitas perguntas, temos muitas discussion. Okay. So to English, thank you. So I want to talk to you about developing creative solutions for averting an agricultural phosphorus crisis. And as Anna said, I'm a soil scientist, a soil chemist. And so therefore, we will probably eventually end up talking about solutions to this problem from the perspective of soils. And I'll tell you why as we go along. It's not advancing. Okay, maybe it's not on. Oh, yeah, that helps. The guy. So it should be at the computer, right? Where I'm going to stand on this side. There we go. Okay. So you all having fun here at Ilum? Yeah? You're learning something? Great. Okay. Do you want? I'll just tell you everyone in advance. No problem. So you can advance, yes. Okay, so let's talk about the crisis. Here's some facts. Phosphorus is an essential chemical element for humans and all of life on Earth, as far as we know. In our DNA, our bones, our ATP, all our metabolism, energy, no, we need phosphorus. Second, rock phosphate mines, rock phosphate, which is mined, these are the main source of economically recoverable phosphorus. Third, 92% of that phosphorus that's mined in the world goes into agriculture. And most of that is for food production. So that we can eat, we can get energy, we can live, we can continue, and other farms on Earth. And there are a number of estimates. The worst case scenarios are 7 years, but they go from 100 to 300 years. We have that much 100 to 300 years supply of phosphorus left, rock phosphate from these mines. Okay. So, I mean, there's phosphorus in the ocean. Maybe there are other ways we can get phosphorus, but we consume a lot of phosphorus and rock phosphate, and 300 years left. And you could say, oh yeah, okay, 300 years. No, I'm not going to be around for sure. Same, but maybe some of you will have children, and then your children will have to start worrying about the 100-year issue, and your children's children's children will have to start worrying about the 300-year issue, right? So the problem is gonna become worse. Go ahead and change it. Next slide. Okay. So, and as the world population continues to increase, and it is, that means more demand for phosphorus. So this is just a curve showing, the upper curve shows the projected demand for phosphorus out to 2070 at the current consumption rates. If we just keep going and try to don't worry about the problem. Don't implement any solutions. Okay. So that's kind of the situation. Next slide. Okay. It's all right. Something happened. Was that you, Anna, or? Okay. We'll see if it works. Let me see if this part works. Okay. So the other part that's pertinent to you particularly and me, because I'm living in Brazil right now, what is important for us is that 90% of the phosphate in the world, this rock phosphate, which goes mostly almost 100%, 92% into agricultural production. 6 countries control 90% of that. Morocco controls 70%. Okay. And then China, South Africa, you can see the bars here. Algeria. Brazil has about 0.4% of that phosphorus. Do you have very much agricultural production in Brazil? Aha, yeah. So in order to maintain that production, you need to have phosphorus. You have to put phosphorus in the soil. Unless, of course, next slide, you can think of some other solution. Let's just solve the problem right here and now. Okay, so how can we solve this problem? What are ways we can solve this phosphorus crisis? This impending phosphorus crisis, meaning at some point in your children's or grandchildren or great-grandchildren or great-great-grandchildren's life, something has to change. Any ideas? Finding other sources. Yeah, as far as we could say right now, phosphorus as an element is very critical for most of our, life has evolved using phosphorus. Mainly, a lot of it is ATP, our energy source. So probably there wouldn't be a substitute in life metabolism, right? So in other words, if we use zinc instead, we wouldn't get the phosphorus. We really need the phosphorus. But there might be other sources of the phosphorus, like, oh, let's go to the ocean and start mining up the seafloor and shells and so forth. But that's right now, that wouldn't be the most economic option, because the mines can go in with heavy equipment and get the rock phosphate and process it. This is a great question. Nitrogen, 70%, 70+% of the atmosphere is nitrogen, dinitrogen gas. And since the Haber-Bosch process, the process high temperature and pressure and catalyst, converting that very stable nitrogen N2 gas into ammonia, that helped with the nitrogen. So we are cycling nitrogen that way, another very important element. But no, it's a mineral element, so we can't get it from a source like the atmosphere. It's not a volatile gaseous source. Let me go ahead. Tá bom.
O fósforo que é ingerido e usado por plantas, pessoas e animais, em algum ponto ele vai sair do organismo e voltar para a natureza. Então deve ter alguma forma de pensar o uso do fósforo de forma sustentável, né?
I need a translator.
So what he's saying is that we ingest phosphorus and then we eliminate phosphorus. So I think he's asking about recycling. Is there any way to recycle the phosphorus that's already been in use in living systems?
And this is already being done. So like, for example, human waste, animal waste, particularly because we produce a lot of animals for meat, because in the world, meat consumption is increasing, in fact. And much of that animal manure is being recycled and put back on the farms. Now, in some places, not necessarily many of the fields in Brazil, but in some places like, well, southern Brazil in Santa Catarina and in Europe and the United States, one of the problems is we don't know how to control that phosphorus as well. And we tend to put excess phosphorus on the soil, and then the excess leaves the soil and gets into water. And phosphorus is often a limiting nutrient in fresh waters. And if you get enough phosphorus and nitrogen, then we have algal blooms, life blooms, or there's a lot of algal blooms, and then the algae die and the microbes consume the algae and consume oxygen and cause oxygen depletion, and then fish die, da 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 da. Okay, but that is one of the solutions, is to reuse. And we'll look at some options that are already being done. Any other ideas? Like stop eating meat, for example. That would be easy. No more churrascos, right? Stop them. It would help for sure. Other. Thanks, Isabella. Okay, let's see. Okay, so here are some. So this paper, Cordell and White, it was kind of a paper, kind of a key paper that got people thinking about the global problem of phosphorus. It's published back in 2014. But this is that same curve I showed you. If we keep having demand, this is the consumption rate of, this is the phosphorus that we would need projected out to 2070. We're here 2022. So you can see the numbers are almost doubling by 2070 to support life, to support human activity, humans. And so we could increase the efficiency of the mining and fertilizer production. This is their estimates of how we could ideally lower the consumption down here. Efficiency in agriculture, that's a big one. Efficiency in the food chain, changing our diets, right, Isabel? Reuse, this is the recycling. Use the crop residues. We harvest the grains, but then put the residues back more efficiently. All of these things are already being done, but we just need to make them better. We need science, we need technology to make it better, way better. And I'll give you examples. Reuse human waste, reuse animal waste from manures. That's a big one because of our animal production. And then this is supply phosphate rock. I can't remember exactly the detail of that, honestly. But let me give you this point about soils and agriculture. When producers put phosphate fertilizers on soils, so let me back up a bit. We have rock phosphate. It's the rock. Grind this up. It contains a mineral called hydroxyapatite, which is quite insoluble. So it's usually not soluble enough to put it on the fields and use it as a fertilizer. So instead, it's reacted with acid, sulfuric acid, or sulfuric and phosphoric acid to make it higher grade, more soluble, so it's better for the plant, but better for delivering the phosphate to the plants. But in fact, when these high grade phosphate fertilizers are applied to a soil, about 30% or less than 30% typically, or less than or equal to 30%, gets to the crop. Where does the rest go? Any ideas? I already said it does not volatilize. Sorry? Yeah, if you put too much on, when you get excess, it goes to the rivers. But that's not really where the main place where the other 65 to 70% goes. Any ideas? It gets bound in the soil. The soil binds it and doesn't let the plant have it. So this is one of the, I think I have a highlight on this. So maybe flip the slide just to see if I have a, yeah. So if we understand, this is 30%, that's 70% inefficiency. If we could, let's say we double that to 60%. Let's say that we currently have 300-year supply of phosphate, rock phosphate. If we double the efficiency, and remember, agriculture consumes 92%. So let's just say it consumes essentially 100%. If we double it from 30 to 60%, if we can get the fertilizers on and get the phosphate from the fertilizer to plant and double that efficiency, then we can extend it to 600 years, right? Gives us a little more time to find solutions to the problems. Shoot, if we could get it to 3 times 90%, then that's almost 1,000 years. Then we might say, okay, somebody's going to come up with something in 1,000 years, even if we can't come up with it in the next hour, the golden solution, right? So we want to this is one of the many ways we could start to address this problem. So let's go to the next slide. So I'm a soil scientist, so I'm going to take the whole problem down to soils and tell you about soils and phosphorus, okay? I still want questions. I want discussion. All right? Okay. What's a soil? Who can tell me? What's a soil? What is a soil? ¿Qué es a solo? Yeah, we all know, right? We all know what a soil is. It's that dirt, that stuff, dirt. Yeah. So we have soil scientists have, like any scientific field, we have specific definitions of any term. So a soil is the unconsolidated material that covers the Earth's surface. Okay, and it supports plant growth. Now, wait, the definition was changed about 7 or 8 years ago. It's the unconsolidated material that covers planetary surfaces, because the soil scientists want to claim the soil on Mars too. We want to expand our field for operating and studying. And in fact, there's soils are coming back from Mars, right? People study soils that have been sampled on Mars. So the biogeochemistry perspective of soils. Let me just tell you, for example, this is, Embrapa. So Embrapa, like in every country, we have agencies that tend to characterize soils and map them based on their characteristics that affect how we manage the soil. So different soils need to be managed in different ways because they have different properties. So this is a map of your soils in Brazil. So you can, at a pretty high scale, so you can look at all the diversity of soils you have in Brazil. So next. So what are soils made of? What elements compose soils? Here's the main ones. Oxygen is most of it. Silicon, aluminum, carbon, iron, calcium, magnesium, potassium, sodium, protons, and then that's like 98%. And then there are other elements: nitrogen, titanium. Here's what we're talking about: phosphorus. Okay. Sulfur, manganese, boron, vanadium, zinc, chromium, nickel, copper. And then we can even look at contaminants. These are lead, cobalt, arsenic, molybdenum, selenium, cadmium, mercury. Why are all these elements in soils? Because these are elements that are in the Earth's crust. And soils were developed by weathering, breaking down of rocks from the crust. And the elements became incorporated into the minerals in the soil. So there are minerals that might have been formed at high temperature and pressure. We call those primary minerals. And then when those minerals are exposed to Earth's surface, low temperatures, low pressures, they're unstable thermodynamically. So they dissolve, and other minerals that are stable at ambient conditions form. And that's what we're interested in. And then the material becomes unconsolidated, particles break down. So we have sand, like at the beach, and then smaller and smaller particles. This is the the soil, and then we have pores in between the particles that water flows through and plant roots grow into. And to get the phosphorus into the roots, the phosphorus has to dissolve into the water. We can only take up these elements. It can only take up the elements from the water in the soil pores, okay? So I show here these elements that have green, either written in green, for example, on here. These are all elements that are essential for life, as we know, for plants. Selenium is essential also for humans. But these are 16 or I think it's 16 elements that plants need to grow productively. Next. So now we get into the details of soil. So we have, in soils, we have minerals, different kinds of minerals, and also solids that are non-crystalline. Maybe you've heard about those. Maybe you study, you know what minerals are. They're crystalline structures, meaning the atoms repeat in space. We have something called organic matter. Organic matter comes from residues, for example, from plants. So after plant roots die or leaves fall on the soil, microbes decompose them. Here are some microbes. They decompose the material, and what forms is some more stable forms of carbon that break down very slowly. Why do microbes do that? For fun? Sorry? For life, to live, yeah. Why are you drinking that Coca-Cola or that Monster or eating a snack right now? You're basically just getting the carbon out of it, and you're inside the body, the respiration, Krebs cycle, for example, respiring, basically oxidizing carbon to CO2, right? You breathe in, you can take oxygen. The oxygen is used to oxidize carbon. Chemically, that's simple. Oxygen plus the reduced carbon goes to CO2. But there's all this complicated biochemistry that does that. So that's what the microbes are doing. They're fighting for the energy resource, which is locked up in the chemical bonds of this reduced carbon that came from the plant. And where did that energy come from? In those plants. Photosynthesis, mostly photosynthesis. The sunlight supplies the energy that helped to reduce carbon dioxide from the air into biomass, and now that took energy for that process to occur. Now that energy can be extracted again from us or from microbes, etc. So they're primary producers. It doesn't always have to be carbon. Some of the early microbes are what are called chemoautotrophs. They use other chemicals to oxidize or reduce, but it's usually redox chemicals that moves energy around in living systems. Okay, so moving forward, because I'm going very slowly, is that okay? I'm going to ignore your timeline. Oh, I think there were some arrows yet. Yeah. So this is how, so everything gets integrated in the soil. This is kind of complicated because you have the organic matter produced, you have all these living organisms. 1 gram of soil, that's like a teaspoon, contains about, I think it's 100 billion different microbes, 100 microbial cell bodies. Okay, a lot, 1 gram. So there's a lot of living life in soils. And so it's decomposing. The organic matter gets intermixed with the minerals on the surfaces. And then ultimately, the soil itself, what gives these different soils their properties depends on what rock they came from, which elements were in the rock, what other minerals were in the rock, how long that rock and that material has weathered. Has it been 100,000 years or 100 million? Right? That defines soil properties. The climate in the tropics, things weather faster because there's a lot more water going through the soils, and there's a lot more higher temperatures. That both of those tend to weather the soils faster. Relief, topography, how particles move down slope or water flows, or water sits and changes redox conditions. And biota, all the the biological life in the soil that are associated with the soil, including plants, affects the end result. Okay, go ahead. So just about phosphate, we have this, it's not real, it's a kind of a, it's a word we use called fixation. You know where one of the most famous places in the world for people starting to realize there's this property of phosphate fixation is? No idea. Take a guess. Sorry? Bacteria? Oh, bacteria. No, but I'm sorry. Geographic. What geographical location in the world? What country? Ah, Brazil. Yeah. You know those red soils you have? So the Cerrado. You know the Cerrado biome? So in the Cerrado back in the 1970s, they said, oh, wow, look at all this land. If you've been through the Cerrado, if you don't look at the agricultural parts, there's nothing growing. Or scrub us. There's a lot growing. Actually, there's a lot, but it's not very productive. So they said, oh, let's just put fertilizer on these soils and go. So, you know, the amount of phosphate we put on soils is about 40 kilograms per hectare. Okay, so they put 40 kilograms on, nothing grows. They found that when you put 600 to 800 kilograms on, then they can get growth. Okay, why? Because the soils you have, the red soils, take up and bind the phosphate, and you have to put on a lot to satisfy. They say satisfy the soil's ability to bind, and then after that you can get normal growth and put more normal rates on. So phosphate fixation, though, so many people have studied this for a century. So let's look at the next part of this slide. A very simple model is, okay, we need the phosphorus to be dissolved to be taken up by the plant. But when the phosphate fertilizer is put on, much of it goes into the soil solids. That's the brown part here. So this is just like a thin section of a soil where it's kind of a millimeter scale. I'm sorry I don't have a scale for it. I should. But you have big pores like this one and smaller pores and lots of little, even smaller, what we call micropores. And basically, when those pores are filled or partially filled with water, the phosphate can dissolve into the water and then the root can take it up. And so that's the solids, but they have something, the term is labile. That means it can be taken up by the solids and then over time released back to the water as the plant removes phosphorus from the soil water, the solids put more in. But there's another thing called non-labile. Somehow the phosphorus that was put on is going into a farm that doesn't go to the water. It's slowly reversible. So as you deplete this, in principle, that should come out, but it takes way longer than the crop. So then it's not available to the crop. So what kind of questions do you ask when you see that diagram that was produced in the 1980s? You're all smart students. I can use Google Translator pretty well. Labia. That's what it says.
It's labium.
Sorry. Does it make sense?
Like,
oh, lipstick. No, it's this labile. What we say in English, labile is just easily converted back into the soil, solid water. The other one is like found somehow.
kind of part of that use of another question. Can we somehow make kind of think that others were?
These are good questions. These are good questions. And so the first question, could we change the soil in some way? Yes, we can manage the soil. We have to figure out ways to manage it so that the phosphorus can be more efficient. But remember, there's 15 other elements that get. So maybe if we make the phosphorus more available, some of the others won't be available. So it's a complex problem. It's not simple. Synthetic soils, if you have like a mine site that you're claiming, people do that. They try to make the soil properties so that they're more favorable for plant growth. So let me give you a perspective on that. How much do you think 1 cubic meter of soil weighs? 1 cubic meter. Just a little. 400 kilograms. That's a good answer. 500, if I say 501, maybe you can. Boom, boom, Ugala, Joy. Come on. 1,200 kilograms. So imagine, let's dig up this hectare, 100 hectares, and put better soil in. Yeah, but I mean, you don't have to go down to 1 meter. You only have to deal with 40 or 50 centimeters or where most of the roots are, right? But anyhow, that's to think of it, to replacing it's a big job. But yeah, we can do things like change the drainage, how the water moves through to hold the water better. We can change the pH. In fact, to get the soils in as hot of productive, they said you got to put a lot of phosphate on, and you have to put a lot of lime. It's say lime, but it's actually calcium carbonate to bring the pH up. Now plants can grow because they get more phosphorus. There's less aluminum. Aluminum is toxic, even though it's the third most abundant element in most soils. Okay, it's very abundant, but it's not toxic. You know what? It goes into the water and binds on the surface. So the plant roots and then disrupts the cells of the surface. And the roots don't grow root hairs right. They become leaky and they don't function as well. Okay. Thank you so much. I got carried away without it. Scratch what I just said. It's not on the record. No, I'm kidding. Okay, so anyhow, moving ahead, let's look at phosphorus, what we were just talking about in the context of the bigger scale and go into some details, because when I asked you the question, we see that the phosphate goes into something we call non-labile. The normal question that we have to ask is, what is that? What is non-labile? How does that work? Or we can say, how does phosphate fixation work in soils? What causes it, right? That's what we do. That's our research world, to go down and try to figure out what causes it. So just to give you some context, just an overview. Phosphorus, here's a crop growing, corn, maize. Here's a soil. And next slide. So the world average content of phosphorus in soils is 800 milligrams per kilogram. Okay, that's like pretty small amount actually. But from our perspective of the total phosphorus, that should be enough to supply a number of years of crop growth if we could just make all that come out and go to the plant. That's the world average. So the next slide. So all we got to do is get the phosphorus from the soil into the plant. Next slide. So this series of slides shows a calculation I made for a book chapter where, and other people have done this calculation, but I said, well, in the top 50 centimeters of soil, and let's say 1 meter cubed, or 1 meter squared, 50 cent, half a meter down. If I take 800 milligrams per kilogram, what's the total amount of phosphorus in that volume of soil? Okay, now how much does the plant take up? How much goes to other sources or other places? So the plant, one crop needs about 0.2 to 0.6% of all the phosphorus that would be in the top 50 centimeters of soil at that world average concentration. Okay, less than 1%. Okay, next slide. So we put phosphorus fertilizers on at a rate of about 0.8 to 2.4% of that total P. So you already see the problem. Let's look at the big numbers. We're putting 2.4% of what's in the soil already. We put that on, and the crop's only going to take up 0.6, right? So that's 25% efficiency, right? 0.6 of 2.4, or 1/4, 25% goes up. The rest goes into the soil. Well, what happens in the soil? Next slide. So, a little detail. We have to get it into the water to get it up into the plant. The root has to take it up from the water. And it doesn't have to be like a swimming pool of water. There's tiny pores and there's thin films of water around the roots. So it just has to transmit through fine micron to millimeter size sheaths or volumes of soil. But there are pores that can hold a lot of water, especially after it rains. So next slide. Oh, by the way, that pore water of the total, it's what's in the water is 0.005 to 0.015%, meaning that most of it's bound in the solids. Okay, next. Okay, so the point is getting it from the solids to the water. And then we can have some losses. Actually, running off is one of the biggest losses, like particles. Here's what happens. The soils bind phosphorus, okay, strongly. Fertilizers are put on. The phosphorus is bound so it doesn't move down with the water very deeply. But then you can have erosion if the topography, if the field or the soil is slanted, it has a slope. And so this can erode particles, and the particles can have bound phosphate, and then they can move off site to the water. And some of it could be dissolved and taken off. There could be what we call leaching, movement through the water, but that soil down, but it doesn't happen so much because the phosphorus is bound so strongly. Next slide. Okay, so what about the solids? Well, here's all the details finally. So we can divide phosphorus into inorganic phosphorus and organic phosphorus. So usually 30 to 40% is organic P and the rest is inorganic P. So let's look at the inorganic P. We can have different minerals, calcium phosphate minerals. There's a number of calcium phosphate minerals, iron phosphate minerals, aluminum phosphate minerals. And then we can have what we call adsorb phases. That is, the phosphate binds on the surfaces of particles of soil. And then that can be on iron oxides, aluminum oxides, and it can also be on iron and aluminum that itself is bound in that organic. Okay, so there's a lot of details to it. Then there's organic P. Where does organic P come? I won't continue until one of you answer that question. From decomposition of material. Yeah, like we got phosphorus in us, right? We need phosphorus, so we have phosphorus in our bones, but we're talking about mostly in the organics, right? In various forms in our bodies, in the microbes, the plant. So initially it's organic. So it can be next slide. It can go from organic to inorganic. Typically the plant takes up H2PO4 or HPO4, those ions. That's the typical form that gets taken up from the plant. So in the solid phase, it could be these various organics. It could be in biomass, it could be in residue, it could be in organic matter, which we sometimes call humus. And then that, if that converts to inorganic and goes to pore water, then it could be taken up. If these minerals dissolve or the phosphate on the surfaces of minerals come off, then it can be taken up. But most of it's in all these forms. So you see the problem's a little complicated. We want to remember, we want to increase the efficiency from 30% or 40% if we want to aim low. 60% if we want to really have our great grandkids be able to have no problem with this phosphorus issue. Okay. Next slide. And then in the pore water itself, there's a lot of, even though there's very small amounts, parts per million levels, there's a lot of chemistry that occurs in the pore water itself. So this is the problem, and many people have worked on this problem of how to understand how phosphate behaves in soils for decades already. Okay, next slide. So now to kind of summarize and put in perspective again, go ahead. I mentioned already that on these soils, when you see soils like this, have you seen these around? Yeah, no, so when you drive by, you're driving along, and this is out by Paulinia, where I was riding my bike one day, and I said, wow, that's a nice profile. You probably saw it. They were building out, they're building out there, but I saw this road, this profile. And this is a deep soil, and all that red are the iron oxides. Remember I said iron oxides tend to bind phosphate. And so in soils like this that have not had phosphorus for a long time and have weathered for hundreds of millions of years, and many most of the composition of the elements composing the soil, many of them have been leached. They've been gone. They're gone now, including phosphorus, calcium, and the minerals that are stable over long periods of time and high leaching are oxides, predominantly iron and aluminum. And they tend to be the minerals that bind a lot of phosphate. So as I mentioned to you in the Cerrado, the first year they put on 800 kilograms of P per hectare, and then go ahead, next animation, the crop takes up about 15 of the 800. Again, the soil binds the rest, and then you have over the long term, you can put annual amounts that are more consistent with what a plant takes up. But it's still, this is 3 times what the plant takes up. So you keep building up phosphate. So if we can do experiments in the laboratory where we take a soil, we can take a sample of soil and we divide it into a bunch of centrifuge tubes, and we put different concentrations of water with phosphate with different concentrations, and we react it for 48 hours, and then we centrifuge and filter out the water, and we measure how much phosphate is in the water, and we can make a curve like this that shows the relationship between the dissolved phosphate and the amount that's bound in the soil. Okay, so these are curves from 2 soils that were done in the laboratory, and if you take, this is in milligrams per kilogram of soil, and if you make some estimations of the bulk density, which we have good estimates for the soil, we can calculate that these highest amounts, which are like the maximum phosphate, correspond to 770 kilograms of P per hectare in that soil and 340 in that soil. And those names are, soil scientists have names. We have a whole classification system for soil. So there's a typic redox, it's an oxisol, we call it, and a typic plinth aquox, which is also an oxisol. But they're pretty consistent in pH. They're both in Brazil, but they have, almost more than a 2-fold difference in phosphate absorption. Again, why? That's always the question you want to ask. Why? Okay, this is something you can do simply do in the laboratory and then say why. But we can also see that this behavior seems to relate to what happens when farmers try to manage the soil. So there's a relationship between the details that are happening in the soil and how it affects food production, phosphate efficiency, etc. Next slide. Okay, so I'm going to just, this is a conceptualization for you. So why the smallest soil particles? I'm a soil chemist. So sometimes we did a lot of experiments with phosphorus and other elements. We take the soil, and the first thing we do is we get rid of all the sand, and we just work with the smallest particles, either what we call silt, which is between 50 and 2 microns, along with clay, which is less than 2 microns, or even just the clay. Why? Why do we throw away the coarse particles? Well, when we're talking about absorption on surfaces, we can justify that, even though it's not completely accurate. And I'll show you why. So imagine we have a sand grain, and I idealize this as a cube because it's easier for you to see the calculation, but it could be a sphere like you find at the beach. And let's say we take that sand grain. A typical sand grain, because of the density of the mineral, would weigh about 2.6 milligrams. Now, if I divide that sand and the surface area, you can, it's 1 millimeter by 1 millimeter by 1 millimeter cube. Okay, so it has 6 faces. So the surface area is 6 millimeters square, right? So I can take that particle and divide it, weather it, like a soil weathers, break it up into 8 particles. And now each face is 0.5 by 0.5 millimeters, split it in half in all 3 directions, right? Now the surface area is doubled to 12 millimeters square. And if I take each of those particles and divide them in 3 dimensions in half again, then I've got 64 particles, and each length is 0.25 millimeters, and the surface area doubled again, 24 millimeters square. And I can keep doing that. 512, divide those, I get 4,096, divide those, and I keep dividing and dividing and dividing and dividing. And I finally get to particles that are 1.95 microns, which is the clay size. Clay is 2 microns and smaller, so it's the biggest clay particles. And I have 134,217,728 of those. And the surface area is 3,072 millimeters square. So 500 times what we started with. Okay. It still weighs 2.6 milligrams. Okay. The same mass. All I did was divide it. So if we're talking about phosphate ions binding on surfaces, then this is probably where the action is. Okay. Next slide. So now let's go look at some minerals. These are soils. Soil samples, see how much phosphate it absorbs. Remember, I said there's 300,000 soils mapped in the world. So we could say, well, let's get these isotherms for each of those 300,000 soils. Well, they're pretty big scale. So if we really wanted to do it right, we'd have to go from field to field. And in fact, from this part of the field and this part of the field, and we'd have to take millions of samples. And can you imagine that? 10 centrifuge tubes, triplicate, putting all the phosphate in, working for 3 days. Okay, I got one done. Only 299,999 left, right? This would get a little boring. So what we try to do in soil science and in any science is to understand fundamentally how things work. So one thing we can do is say, well, we know the soils have minerals in it. So let's look at the minerals from the soil. We can actually, and we did that a lot. We synthesized the minerals. Okay, and then we study them and we say, well, this soil has 10% of that mineral and this one has 5%. So now we can start to understand other properties that can be measured of the soils and understand how those minerals influence it. So from these isotherms, we call these adsorption isotherms because they're done at constant temperature and actually constant pH. And so from these, we can get what happens. It curves off and it comes to it levels off. Why does that happen? Any ideas? Here's my surface. Here's this table. Let's say that I could put sites on this table. I'll just put X's where phosphate can bind. Okay. Let's say on this table, I have 100 X's. That's where phosphate, we're at completely different scales, but it's conceptual. Okay. Phosphate can bind and one phosphate ion on each of those X's. When I get to 100, what happens to number 101? there's no site for it anymore, right? I've reached the capacity. So this is how we think the minerals work when they absorb. They have these finite sites, and then the phosphate binds. And as it starts to approach, you put more and more in. And there's little details like, oh, if there's a phosphate on this site, it's harder for a phosphate to go on this side, or it comes off easier. So the solubility curve, it becomes more soluble as you put more on, and then you reach a maximum. That's how the explanation for this isotherm. That makes sense? Okay. So next slide. So now we can take measurements of that maximum for different minerals. And let's just put the animation on this one. Here are some of the key minerals that are found in these soils of Brazil that absorb so much phosphate. Kaolinite, it's an aluminosilicate, so it absorbs about around 20 to 30 millimoles per kilogram of phosphate. Goethite, around 200, that's 10 times more, but we have a lot less of it in. Gibbsite, maybe 300. And then we can have non-crystalline forms. So remember I said that the organic matter gets on the surfaces, and it changes the minerals, and the minerals can dissolve, and then when they reprecipitate with all this organic matter around, they don't crystallize as much. So these poorly crystalline minerals can absorb. Look, we're getting up to 1200. We routinely synthesize this ferrihydride, and our initial, when we synthesize it, around 1800 millimoles per kilogram. Okay, so now if I know how much of each of these minerals is in the soil, I can phosphate it would absorb, right? Wrong. You do that and you add it up, and it doesn't always add up. So there's even more stuff going on that we don't understand. And moreover, everything's changing over time. So we lose phosphate. There's some other reason that the phosphate disappears, but it's a good first approximation. And you also see what we're doing. We're breaking the system into more detail and trying to understand the details of the system. This is what we do. This is how we study these systems. Next slide. So here is this molecular structure of a mineral, iron oxyhydroxide, goethite. It's what makes soils yellowish. The yellowish color in soils is goethite. The reddish color is another iron oxide mineral called hematite, Fe2O3. The darker brownish colors are organic matter, typically. So then the mix of all of those is some color, that's some mix of all of those, okay? And then the things like kaolinite, they're grayish and whitish, and other minerals, quartz is clear, you know, quartz crystals, or they could be milky, they could be whitish colors. So these are the kinds of minerals that give the soil the colors that you see. Okay, so if you add it, how does phosphate bind? On this mineral surface, so this is just a tiny piece of the mineral, bigger, but it's still less than 2 microns, usually in the hundreds of nanometer size in soils. So it can absorb with 1 oxygen of phosphate. Basically what happened on that animation, it replaces this oxygen. So this is an iron, and this is an oxygen, and these 2 little black dots like Mickey Mouse at Disneyland, these are protons. So that's FeOOH. Okay, so you have some of the oxygens have protons, but not all of them. So phosphate can bind with one of its oxygens, replacing that oxygen and forming a covalent bond with the partially covalent bond with the oxygen. That binds it pretty strong. We call that, we have names for these in communicative science. Monodentate, meaning one bond, one tooth, one grab. Inner sphere, it means that there's no water in between. It's not electrostatic. It's actually interacting with the iron, the oxygen from the phosphorus, covalently, partially covalently bonded to the iron. Surface complex is on the surface of a mineral. So that's a name we give it. Next animation. So we can have 2 oxygens binding 2 irons. That's bidentate binuclear inner sphere surface complexation. So now we have a stronger bond. To get that phosphate off, we have to break 2 bonds. So the conventional wisdom, you know what that is? What Todo Mundo thinks that this, because because you see red soils in Brazil and you see lots of these oxides. I mean, it has lots of these oxides and it finds a lot of phosphate and it fixes phosphate. That it's because of this. Okay, we can just predict it from the mineral. Maybe that's correct, maybe not. These are pretty complicated soils to study. So then there's one more possibility, which is called a bidentate mononuclear. So it's two oxygens binding to one ion, okay? So there's different ways. So you see what we're doing? We're getting into more detail. Isn't this fun? Somewhere up there, we got to solve the 300-year problem, right? But to get there, we have to figure out what the heck is happening first, and then so we use different tools. I heard you have a really great lab here. You have lots of nice equipment to study things, right? Yeah, you can study all of this and come up with a solution to this 300-year problem. Next slide. So this is one hypothesis. This is the same slide I just showed you, but we can also look at the surface configuration of the atoms on the surface and different surfaces of a mineral, the way the atoms are configured, find the phosphate differently. So those are details at a molecular level, what I just showed you. I call it their central dogma. Then you have a very famous soil phosphorus chemist, Jim Barrow, says, no, there's defects in the structure and it has to be a diffusion process. So the phosphate diffuses into the minerals, okay, and gets inside. And then the minerals kind of crystallize over, so the phosphate's inside and can't come out easily. Then next one, we hypothesize that, well, if that can happen, then when we have a soil, like this is a 1 cubic micron schematic of soil with different minerals, and you have pores and all kinds of surfaces of different ones. It's more complex than just one mineral, that the phosphate can diffuse into nanopores of what we call microaggregates. So that's one of the things we're studying over at LNLS right now. And then, but that came because there was a paper published in 2021 by Smolder's group that that they studied how phosphate comes out of a bunch of soil samples, like the isotherm, but they put soil in water that had phosphate and just watched how it came out for them. Or at least the long-term months. And they hypothesized that the phosphate can go into the pores or be blocked by organic matter that blocks the pores. So there's a lot of physical details that could be happening about the phenomenon that could happen in soils. And so, by the way, we have, if you go to the next animation, we have lots of tools that we can use over at Sirius and at CNPEM in general, like beam lines. I know you've been to Sirius more than once, right? So we have beam lines that can study how the surfaces, how the phosphate binds on surfaces like Sabia and Carnauba, and then how the structures, Pinera is X-ray diffraction, the crystallinity of the minerals, and again, these beam lines can be used. Carnauba, Imbui, which is an infrared beam line. Carnauba, Catarete. We're using Carnauba, Catarete, and Imbui to understand how phosphate, ultimately how phosphate, we're really just looking at the structure of microaggregates now. You have a question? No? Okay. And then we can look on a bigger scale at Magno, which is tomography in Carnauba. Okay, next slide. Okay, and then also, that's just, I've been talking about soils recently, right? Because I'm a soil scientist. What else is in this, the whole? We have the soil, and we're trying to get phosphorus to plants, right? So then what else do we have to consider? The plants, yeah. And what part of the plant is down in the soil? The hyzas, yeah, the roots. So I'm sorry, I am not talking to this at all. So this is one of the this is an animation from some X-ray tomography work that we did at Magno a couple years ago. It just got published actually, and it shows two roots in soil. So we want to understand how the root grows and how it can grow in relation to the fertilizer particles. So maybe we can just develop a, go ahead, question. Yeah. Well, this is an example. This is, I'm not showing a lot of our data here because I think this is being broadcasted to the world, right? So, but this is something that we published already where we were just looking at how roots grow in soil. And then we want to understand why roots can grow different places. And if let's say we put a phosphate fertilizer particle, we want to get the phosphate to the root. So if we put a phosphate fertilizer particle here in a soil, how does the root grow? Will it grow to that? Does the root grow something, do something when it gets near? Does it sense the phosphorus there and then change how it grows and the direction it grows to be able to take that phosphate up? How can we move that phosphate? Maybe through nanoparticles to the root, for example. So going back, if I can go back one slide, here it's surface chemistry. So these beam lines can look at phosphate and how phosphate is bound on surfaces. We use spectroscopy. Okay, we get X-ray spectroscopy. We change the energy and we get spectrum. And the way the phosphate is configured on different surfaces gives a different spectrum. So we can say on this mineral with this kind of surface, phosphate's binding in this way. That's correct. These are beam lines here. These are the names of beam lines. This one's a soft X-ray beam line, so soft, low energy beam line. And basically it's at energies that can excite electrons out of the L shells of phosphate ions. And that gives a spectrum that can tell us if it's bound with calcium or iron in a certain way, or aluminum. And this one is a hard X-ray beam line, which is higher energy. So if we went to 2150 electron volts, we can excite electrons, the core electrons, the ones right next to the nucleus, the S electrons, and we call that the K shell. So we can get K-edge spectra. And a lot of people have already done this. We've been doing this for years. We published a paper in 2003. It was like the first paper using that technique to say how phosphate is bound in an actual soil, in actual soils. That was in 2003. So it's not new, this is being done. But what we're doing here, for example, we're taking 20 micron micro aggregates out of soils, and then we're doing nanotomography to look at the pore structure. And we'd like to understand and model how phosphate diffuses in. And so you're imagining what you can imagine from this picture is if I have iron oxides, the yellow ones, and let's say aluminum oxide, the white ones, and organic matter, those dark blobs, and they're all mixed together. And a phosphate ion goes in. Well, it's going to absorb on the iron oxide differently than the aluminum oxide or the organic matter. We want to understand how that works. And if it goes in there, and what we're finding is there's fungi in there. Okay, so there's biology going on, and there's the possibility. I mentioned that mostly redox chemistry is driving how we get energy. So there are microbes that instead of using oxygen to oxidize carbon, they can use iron to oxidize carbon, iron oxides, and the iron oxide gets reduced. And if iron oxide, which is very insoluble, after millions of years, it's still in the soil and given the red and yellow colors. But if it gets reduced, when the oxygen can't get into those microaggregates fast enough, it gets reduced from iron 3+ to iron 2+. It becomes very soluble and it can move around. And then as soon as any oxygen hits it, like when the water drains out of the soil over time as it's drying, then it reprecipitates. So we think it's possible that the phosphate migrates into these nanopores and micropores, and that the iron can move around and then reprecipitate and block the pores. That might be a mechanism that contributes to this fixation. Does that make sense? the soil bind the phosphate, but instead somehow between the fertilizer, the soil, and the root, get the P to the root more efficiently. So I can take that 300 years out to 600 years or 1,000. Okay. And temporal scales, because we're talking about the fusion and processes that occur over time. And I just talked about. We got other problems. Did you notice? Carbon in the atmosphere, for example. There's lots of problems that soils are important for and that technology can be used for. And so, you know, we also have a lot of phosphorus in soil, so we want to know, how do we recover that? Okay. And this was from a pre-proposal. Anna was involved with it a couple of years ago now. They finally just picked the one that was funded, as you know, but we were planning a large proposal to work on this with we had 40 scientists on this from different disciplines to look at all aspects. This is you, in my opinion. We want to empower and impassion you. I want you to be excited about soils and about this problem and to go solve it. Because I care about my kids and their kids and grandkids and great grandkids too. And then we need to be able to take what we learn down here at these scales and actually scale it up. That's not an easy problem to use it on the, you know, to manage the soil in a way that we can improve. So I'm really fortunate, the next slide, because at CNPEM, I work with a whole bunch of people. And I'm sorry I don't have you on this slide. She's part of it too. Or you know, I think she's got you looking at glomalin, right? Yeah, oh, glomalin. Yeah, so all this stuff is that we're thinking that these microaggregates we studied at the fungi that release glomalin are actually creating those. That's everything we see. It looks like these fungi are creating these microaggregates, and I told you about how they could be important. So we need to understand these processes. But there's a lot of great people that I'm lucky enough to work out at LNLS and CNPEM in general. And then that's it. Next slide and last slide. Thank you. How am I doing for time? Okay.
Aquele primeiro esquema que você mostrou com o dogma central, tem uma desidratação naquilo, eu acho que eu não entendi muito bem. A partículazinha de água acaba saindo quando faz a ligação do fósforo?
She asked when you show the slide, the figure of the central dogma, tinha when phosphorus binds in that central dogma schematics, you lose water, so the water molecule comes out whenever the soil comes in. Você perguntou se a água sai, então é isso mesmo. Você quer saber como acontece? É isso? Here.
We can write it as a very general reaction, okay? So we can have, let's say FeOH, okay? So when the iron's not going to be at the surface, there's so much oxygen in water that the iron will always have an oxygen or either an OH or a water molecule bound to it. It's bound at the surface. So we can have FeOH, and this is solid. This means that we're looking at 1 mole of those sites with the iron and the oxygen. If we could go back, can we go back a couple of slides? Sorry, just so we can, probably about 5 actually, to the one where, yeah, this is what you're talking about. If you go to the previous one, it's that one scaled up. So we're talking about one of these sites like this, say this iron with this O and two, it's actually OH2, but some of them only have one proton on. So I'm writing that, I can write a reaction like I'm looking at one of those sites or 1 mole of those sites, okay? So it's the Fe, and this just means it's on a surface, that little symbol. Here's the OH, and it's solid phase, okay? Plus H2PO4 minus. Okay, what's gonna happen with this? Okay, we have to write a balanced reaction, right? So we're going to keep the site because we're going to bind the phosphate on it. And it's going to be OPO4. And there's probably going to be 2 H's on it. Okay. And in that case, it would be, and this would be now the phosphate's in the solid, and here it was in the soil water. And then it would be plus an OH minus aqueous. So if I bind that phosphate, the pH would go up. I could also have this with, I can write the same reaction. I can add a proton there, or at certain pHs, these surfaces, if you go down lower the pH, there's more protons. Another proton will bind on the surface, and it's basically like a water molecule on the surface. So we can write the same reaction, but with FeOH Fe2 plus H2PO4 minus goes to FeOPO3 plus H2O liquid. Okay, so let's see, I have to make sure I've got this, yeah, charge balance. I got a negative and 1 negative. That's fine. This is no charge. So then we have to have, we need a negative charge, don't we? So we lost a proton. Oh, no, I'm sorry. We have the H2, we have the H2PO4, okay? So we needed 1 more proton, I believe, plus a proton. I have to look at that in more detail, but anyhow, that's how we write the reaction. So basically the phosphate is displacing that oxygen and binding directly to the iron with its oxygen. And phosphate itself is covalently bonded, so the phosphate is going to stay together. Okay, you're welcome.
Hi, Dean. Thank you so much
Hey.
for being here. Thank you for your lecture. And since we don't have so much time, I'll try to represent all the second class here, because we've been studying glomalin for the whole semester. We even had Sarah Adrian here to give us a lecture. Sarah
Oh,
Adrian,
yeah. Okay.
she
Great.
came here.
Nice.
So we were really curious to know what you have to say about glomalin, since you showed that there's a lot of evidence that in the pores of the microaggregates there are probably hyphae, and glomalin is a protein produced by hyphae, right? So we wanted to know, like, how could glomalin influence all this soil structure, and maybe even like the iron that is present in the microaggregates. We wanted to know what you have to say about that.
Yeah. By the way, please fast forward this to the second last slide with all the people.
Yeah.
That one back, yeah. Where are you, Isabella? There you are.
Yeah.
There you are, and you're someplace else in here on the infrared, I think.
Yeah, I'm sieving soil there.
That's how you're sieving soil, yeah.
So fun.
So yeah, Isabella was involved in working with us for a few weeks, and the glomalin is a cementing agent. It tends to stick the particles together. We're looking at all these particles, and since you were there, I can show you some images later. It's amazing some of the things we see because we made the sample different ways, but it seems that the hyphae, they're exuding this probably glomalin and other polysaccharides that just tend to cause the particles to agglomerate together. And we aren't sure if it's just because we see particles where you can see, and you remember the structures with appendages sticking out, and we think, and we were working on the evidence that inside all those appendages, those were hyphae, and then particles are coming around, and the glomalin must be gluing it together. And then that keeps the microaggregate intact because, as you recall, we took the soil and we shook it very hard for 2 hours at a high rate. And we collected the particles that didn't break apart. And so over time, there's many people think that these microaggregates are stable for decades, for years to decades. So we don't know the whole process. It would be nice to follow and understand that, but we feel like what you're studying, that those molecules that are exuded by fungi could have a significant role in forming these, what we call secondary structure. The primary particles agglomerate into secondary particles. Thank you. Other
Mais
questions?
perguntas, gente? Bora. Uma aqui. Vem aqui para te ver.
Hi, Dean.
Hey.
The excess of phosphorus can be harmful to mycorrhizae. Mycorrhizae.
Mycorrhizal
Yeah.
fungi.
Yes, because it affects the mutualistic relationship between the plants and fungi. Does this type of problem really affect the soil culture, or?
Yeah, is it affecting what we do when we're plowing and doing all that? Well, as you probably saw from Sarah, if Sarah gave a lecture, she might have talked about mycorrhiza fungi too. And yeah, this is a really great question to be solved. I don't know the answer directly, but I know that, and Sarah probably already told you that if the plant root can get phosphorus itself, it's not going to give up its carbon, which is an energy source, to the fungi to do it. There's a person in the Netherlands named Toby Keers that studies this in great detail. And they look at how different fungi try to trick the plant. Like they'll bring phosphorus, but apparently they move it in, and I learned some of this from Sarah, in polyphosphates, but the plant root will need phosphorus, so they move it as polyphosphate. It breaks down into orthophosphate, just single phosphate ions at the root, and then that's what the plant would take in the arbuscule, I guess, the interact where this symbiosis occurs. But some of the fungi bring the phosphorus, and the plant thinks it's going to get it, but then they don't break it down, or they take the carbon and they do things to get as much energy and as much resource for themselves. So it's maybe not, as Toby Keir says, it's not as symbiotic as it could be. She has some theory called biological market theory. That's like a market, so you might read those papers, but there's a lot of detail about that. I can't answer directly, but I hope that gives you kind of an idea of what you need to look at, or what could be looked at to try to understand these details. I think that if there's a farmer sitting on his tractor plowing the field, he's probably not thinking about that, right? And the other thing is, microbes are very prolific. As soon as they have an opportunity to grow and get energy, they will. And remember, there's hundreds of billions of these in small amounts. So things can always regenerate, the microbes. Welcome.
Não tem mais pergunta, então depois. So they're a bit shy.
Oh, that's fine. Do any of you, how many of you are interested in studying soils? Besides Isabella? You? So only one. Ah, I thought I'd hear like
Which
50
one?
of you.
There's one raising their arm.
Oh, quem de vocês quer solos estudar?
Quem quer estudar solos? Quem gostou da...
Ah, Yeah, okay.
Ah,
Okay, yeah, Kim goes to solos. It's muito importante para o Brasil.
então, agora,
Solos são muito importantes. E eu gosto que as pessoas aqui, vocês, e os pesquisadores que eu trabalho, appreciate soil. In the U.S., in the United States, os Estados Unidos, eu say, oh, cientista de solo. Serious. People ask that. What is soil? You can study soil, but here people really appreciate it. It's great because it's one of your very important resources, natural resources.
so now the very last question.
Yeah, it is a very difficult one. I mean, it's a classic question. When you was a children, what do you wanted to be when you grew up?
A professional baseball player
Or
or an astronaut.
and no, okay. This part I don't want to. I would ask to you if you did realize this, but astronaut, I don't think you are astronaut. I'm sorry, but as a professional,
So I was not good enough at baseball to even be an amateur baseball player. Okay. And there aren't many astronauts in the world. So at some point I started studying then I wanted to be an engineer. So I started studying engineering, but at that time, math, I thought, oh, this is I didn't understand this. I didn't get this math stuff. So I changed to be an ecologist. I want to save the planet, right? I want to study ecology, how the world works, how natural systems work. And they didn't have that as a major. So I started designing the major. And it's interesting because I grew up in a rural small town of 100 people in the Midwestern United States with farms. I worked on farms. My family had farms, but I didn't want to do any of that. And I took a soil fertility class, and what struck me and got me interested, and the instructor was talking about when a single fertilizer granule drops on the soil, all the chemistry, the pH goes up and then it goes down. And I was like, this is amazing. And that's how I said, I want to study this.
okay, I want to be exactly as you are when I grow up.
Well, you're growing up.
joking.
We can talk about that anytime. I'm over at LNLS, and if you want to talk about being a soil scientist, you let me know.
Okay, thank
All right, great. What's your name?
you. Gustavo.
Gustavo.
Okay,
Dean.
Dean.
Pleasure.
It's a pleasure.
So let's thank him again. And
Thank you all. This was really fun. I really enjoyed it. Seriously.
now you all go to the English class. They have English class now. So
And I've taken Portuguese too. So maybe in a couple years I lecture in Portuguese. Hey,
Nelson.
Hi, Nelson. I'm good. Good to meet you.
Very good talk.
Thank you. Are you an instructor here, I guess?
Yes.
Okay, great. Oh, okay. Oh, you're the one that we interact.
Yes.
Thank you. Sorry about the abstract. I got late on it. It just went off my radar. So. Okay. All right. Thank you so much, Okay.
You're gonna get a lot of engagement.
And I hope it gets people interested in
Yeah.
studying these things, which is fantastic. Thank you so much. My pleasure. I thought it would be more like a discussion, and then every now and then I have a slide.
I think actually Adam's question for hearing that many of them they don't speak English and
Yeah.
maybe we're not understanding what we're being
And also
told.
the time, I just ignored the time. Sorry.